everybody. Welcome to yet another episode of Health Discourse with Sakshi and Sushant. So, hi Sushant, how are you doing today?
Very well, good morning.
Good morning. And today, folks, we will be discussing about the role of mitochondria in our overall health. So, as all of us know that, mitochondria is something that you must have studied in your biology classes during your ninth and 10th grade, and where you told that it is it is a small organelle of a cell which plays an essential role in energy production. Okay. However, when we have been researching at Gene Fitletics, we came to know that it is more than that and it holds an extreme importance to our health. So, Sushant, would you like to start the discussion by elaborating that
Mm.
why is it so much important for human health and how it is connected with the development of any kind of diseases?
Okay, so first of all, let me correct you. Mitochondria does not produce energy, it transforms energy, right? So,
Correct.
if you go basics of physics, law of thermodynamics, energy cannot be created or destroyed, it can only be transformed. So, essentially, since you mentioned that, we all know that mitochondria is the powerhouse, right? And it produces ATP, right? So it transforms the energy. So ATP is nothing but transformation of the biochemical energy into electrochemical energy. So there are electrons from the food which gets into our body when we are consuming food, right? And those electrons flow inside the electron transport chain or the respiratory chain of the mitochondria, right? And then at different stages there is a resistance, there is a stoppage, right? And this stoppage is what is essential for you know energy transformation. So consider this, if there's a flow of electrons like electricity flowing directly on you know on oxygen, so probably it's on candle, right? So essentially it burns into flames, it becomes it combusts, right? So essentially the electrons meet oxygen in the complex for complex 5, the electron transport chain, and that's how ATP is actually, we get ATP, and ATP fuels various biochemical process inside our body, right? And this is, and that the basic difference between a dead person, a cadaver, and a living person is the flow of energy. It is not about genes, it's not about molecules. So different. So if you have to sequence genome of a person who is expired, who is dead, and and when he was alive, you will not find any difference, right? So ideally, it is the flow of energy that defines, and mitochondria is, that is one of the functions. The transformation of energy is one of the functions of mitochondria, right? So mitochondria, what we found and what we now also measure for our patients is that mitochondria is the motherboard of our cells. In fact, the genes or epigenetic modification is driven by mitochondria. So it's like a distributed network. So there are mitochondria in every cell except the red blood cells, right? So whether it's brain, heart, lungs, liver, pancreas, kidneys, right? Every cell has mitochondria, right? Someone has more mitochondria, someone has less mitochondria, right? And again, those mitochondria speak to each other, so there is an distributed network. They communicate what is going on, and then based on that they actually can produce heat, light, electricity, or even trigger, turn on or turn off a specific gene that leads to production of cytokines and other mitokines and metabokines, and that drives the cellular adaptations and the cell to cell communication. Right? So, mitochondria such have all the receptors. So, in fact, it can sense the environmental cues and make specific, trigger production of specific cytokines that could, you know, uh, communicate the current state of the health right now, right? And that's how there is a reallocation of energy. So going by law of thermodynamics, and since there is an limited capacity to consume food, so there isn't you know a daily energy budget what we have, right? And that budget is allocated between different competing uh, activities, right? Like some is mostly is devoted to or allocated to the essential functions needed to live, like breathing, like heartbeat, blood flow, right? How our neurons are functioning, right? And then a part is given is allocated for growth, maintenance, and repair, right? So our cells undergo a lot of assault and encounter a lot of toxins, and those things lead to sort of a lot of uh, you know dysfunctional cells, and we can repair and replace dysfunctional cells, or we can drive the growth of our cells, right? Growth of our body, and so all those needs energy. And again, and then third part is obviously our response to different stressors, right? Whether it is environmental stress, whether it is stress coming from food or microbiome driven stress, right? So again, there is an allocation of energy, and when we have a lot of these stresses, those stress actually energy is relocated from growth, maintenance, and repair to response to those stressors, and then our body shift to a new adaptive state, right? So disease is nothing but a new adaptive state, a new allostatic state, right? So your body is trying to keep you in a homeostatic position, and then they move to a different state altogether, right? And again, if the stresses are not, you know, do not take actions to fix those stressors or to prevent those inflammatory metabolites and stressors from impacting our mitochondria, we actually we move from a transitory to a permanent allostatic state, and then we have cellular recalibrations. We have systemic dysregulation, we have a breakdown in the communication cell to cell communication network and regulatory network, and then we have disease. So it's all disease is nothing but an adaptation to a different, maybe inappropriate uh, you know stressors encountered by our mitochondria. And as I mentioned, mitochondria have receptors, they can sense, and then they can take actions, right? Yeah.
Yeah, that's pretty much it's very interesting to know that it plays such a massive role because so far we already knew, of course, energy. The truth is, yes, the energy can never be destroyed. It can only be transformed, cannot be neither be created. That's correct, right? Now, my next question to you is that what causes oxidative stress?
Okay. So let us understand this, that as I mentioned about the flow of electrons. So
Hmm.
ideally, mathematically, we calculate energy resistance, right? Which is varying depending upon our exposure to environment and other our food and dietary choices and microbiome, you know, drugs and whatever, and sleep and a lot of these lifestyle things, right? So there's a flow of electrons. So what we see is there's a supply of electron, what we call is energy potential, which is coming from the food we are consuming. And then there's flux of electron, flow of electron, the electron transport chain. Again, flux of electrons can be high or low depending upon various stressors. So we have to maintain a optimal energy resistance, so that if you have low, it cannot energy cannot be transformed. It is high, it creates a lot of flow back of electrons. So if there's high energy stresses, it leads to flow back of electrons, right? And electrons spill out or leak out, right? And then they fall on the molecule of oxygen, and this is what causes production of superoxide or reactive oxygen species, ROS, right? So ideally, what happens is when there's high energy resistance, high stressors, the mitochondria expands, right? And the molecules which are, you know, passing on those electrons. So ideally, consider as like a relay race, right? So you're passing on, a stick, and you're running, and your ultimate goal is to get into oxygen to produce to give ATP, right? So ideally, there is a lot of these resistance. So probably these molecules also expand. They go, and then they become reactive oxygen species. And then there's more, otherwise there's low ATP, and this further leads to you know low ROS. So ideally, what happens is that we all know about NAD, right? So NAD takes the electron, forms NADH, becomes NADH, then gives the electron to the complex one of the electron transport chain. Again, it becomes NAD plus. So ideally, when there's high ROS or high energy resistance, which could be because of microbiome-derived inflammation or environmental stress such as glyphosate or heavy metals or probably mold toxicity, right? Or maybe disruptive sleep patterns, right? All these can directly lead to high energy resistance, and yet there's high ROS, right? And which leads to low ATP for the high ROS, right? And again, there are ways to mitigate it. So they only, uh, I think two or three antioxidants that can get inside the mitochondrial inner membrane, right? So mitochondria has outer membrane, inner membrane, right? So one is superoxide dismutase, other is glutathione, right? Third is chlorophyll. So all three, these are the only three that get inside the mitochondria to neutralize this superoxide production. So ideally, ROS is again a way of signaling mechanism for the mitochondria, right, to communicate. But overproduction of ROS is something which is really, really harmful for our biology because yeah, one of the aspect is obviously when there's high ROS, it leads to oxidation of tetrahydrobiopterin, which is one of the cofactors of production of nitric oxide. So it leads to functional loss of nitric oxide, and then you have diabetes and cardiovascular disease, sexual dysfunction, and a number of those things.
Apparently, I think it's quite relative. The all these functions that you've just mentioned, they are quite relative to the oxidative stress part and the
Mhm.
manifestation of diseases part, right? Now, my next question to you is that, I mean, since decades, people have been focusing on the genome sequencing for health.
Mhm.
They
Mhm.
say that DNA is the destiny and all that, those things. But, we at Gene Fitletics, we've quite been skeptical about that statement because, what we have found that obviously you just mentioned 10 minutes ago that genes, they do not change, right? Something which
Mhm.
is static can never change, right? Now when we are talking about mitochondria here, so why don't you just let our audiences know that why one should prefer mitochondria over the genomic sequencing?
Okay, so let me blend this. I will keep it short right now, but if you're discussing about cancer, I will connect with that, right? So
Yeah.
ideally, mitochondria is actually, what happens is in response to stressors, mitochondria send signal to the nucleus, right?
Mm,
So, and then it leads to turn on and turn off a specific gene that direct epigenetic modifications, right? And that leads to production of cytokines like, and cytokines and metibokines, for instance, GDF15, growth differential factor 15, which is released by mitochondria of every cell. In fact, mitochondria can leak its own DNA, right? So it can leak its own DNA, which can actually travel to cytoplasm and get inside the nucleus. So if you have to sequence your genome and you find mitochondrial DNA in the genome, right, it is an indication that your mitochondria is under stress,
okay.
right? So ideally, if you're talking of mutations or epigenetic modifications, those are directly a consequence of the mitochondrial
Mm,
interaction with the environmental and the external environment, right?
mm,
So external environment means anything which is outside of that mitochondria. It could be your gut oral microbiome is producing certain metabolites like lipopolysaccharides or precursor or TMAO or a lot of these things, which can
mm,
stress mitochondria, and then it could lead to high energy resistance, and then the production of ROS, or probably they send signal to so
mm,
to nucleus, and then nucleus, they're changing genes, and then there's production of cytokines and production of GDA15 that GDA15 actually all these cells except the brain cell produce GDF15, and brain cell actually have a GDF15 receptor, right? So
mm,
that receptor is actually then neurons actually when they have these then GDF15 when attached to these receptors, neurons will act and they could uh, activate immune cells like microglia cells in the brain, and that could trigger neuroinflammation. So essentially,
mm,
if you have depression, anxiety, brain fog, fatigue, right, headache, aggressive behavior, all these may not be a brain condition, maybe a metabolic condition, maybe a kidney condition, maybe a heart condition, maybe a mouth condition. And because
mm,
those are activated, or those phenotype show up when there is a lot of GD15 being traveling from the mitochondrial cells via blood to actually this particular receptor, which we call as
mm,
GFR receptor. So ideally, this is something which, and today I was reading a paper where it mentioned that clearly that if you are using drugs to inhibit GD15 activity, it would have a detrimental impact on your overall health, because
mm,
GD15 is an adaptive response to stressors, and the idea is to
mm,
fix those stressors so that it doesn't occur again, rather than just trying to manage the symptoms. Right? So again,
mm,
GD15 is also a signal that your energy. So when GD15 is actually your brain received it, so essentially it is a signal for conservation of energy, right? So it is again the same concept of reallocation of energy, so that we have to conserve energy so that we can allocate the energy to deal with these stressors. So that energy is again your immune cells would mitochondrial immune cells will need that energy to actually neutralize these toxins and these harmful pathogens, right? So ideally,
mm,
that is the reallocation of energy happens, right? And if it is persistent, obviously, there will be an overactivation of this, and there will be an overactive immune cells, and this will trigger inflammation, and then, it will steal all the energy from the growth and maintenance, and it will go to the allostatic responses, and then over time you have systemic breakdown, you have disease, right?
mm.
So genome is
That's
is
correct.
is so in fact, since I'll just share about this because that will get too much complex. But in fact, there are certain bioelectric patterns or bioelectric communication networks which can override these genetic instructions as well.
Okay. Well, that's pretty interesting because, I must say that I think all those who have been working on the genome aspect, the human genes aspect, I think they really need to relook
See,
at
see,
how mitochondria is
I'll tell you very,
impacting.
I'll tell you a very simple thing here. So genome is actually they have standard operating procedure, standard instructions. So genome code for
Hmm,
proteins, right? So
hmm,
genome
hmm,
does not know where your hand is, where your eyes is, where your legs is, right? So they do not define the anatomical structure, right? It is
hmm,
actually stored memories in the bioelectric patterns. So essentially, you have
hmm,
to understand that our cells work on electricity. So there isn't. This is something which a lot of people would know that. I'm not sure whether everyone knows about this or not. That our cells have a transmembrane potential. We have to maintain a transmembrane potential of -70, right? And that is something, that is what we call it. You have to maintain that voltage potential of the cells, which is again, the bioelectric patterns or the voltage gradient actually controls those, or is defines this variation in the transmembrane potential, right?
hmm,
So again,
hmm, hmm,
the genome is a hardware, right? And these bioelectric patterns is software. So they define the
hmm,
development, regeneration, memory, and a lot of these things, right?
hmm,
Even situations such as cancer, diabetes is actually driven by your bioelectric patterns, right? So they are. Uh, here the mitochondrial role is more important because the these bioelectric patterns or the these voltage gradients or voltage potential is actually regulated by movement of ion channels, right? So we have potassium channels, we have calcium channels, we have sodium channels, and these channels are found in the inner and the outer membrane of the mitochondria. So mitochondria controls the calcium circuits, right? So ideally,
hmm,
these mitochondria directly modulates these voltage gradients. So
hmm,
again, when these voltage gradients is positive, what is depolarized, it is nothing but cancer, right?
hmm,
So ideally, these voltage gradients can actually, if we alter this voltage gradient, we can alter the anatomical structure. They can even override the genetic instructions,
hmm, hmm.
right?
That's correct. So, Sushant, now let's talk about the part where we can fix the mitochondria? I mean, my question to you is there a way one can fix the damaged mitochondria, and if there's some, you know, imbalance going on around in the functions of the
There's,
mitochondria?
there's
So
no
is
there's
it
no damage to mitochondria. So see, you have to
okay?
understand that mitochondria, there's an energetic process of the mitochondria, right? So
Okay.
transforming energy, communication with
Mm,
nucleus, communication with other mitochondria, uh,
mm,
making stress hormones. So cortisol is actually made inside the mitochondria, right?
mm, mm,
The cholesterol from endoplasmic reticulum to mitochondria is transferred because mitochondria uses cholesterol to make steroid hormones such as pregnenolone, right, which is a precursor for most of the hormones like estrogen and testosterone. Right? So mitochondria
mm,
plays a very important role. Similarly, if you
mm,
talk of pancreatic beta cells, it releases insulin. For that, it is a very energy consuming activity. So pancreatic beta cells need a lot of energy to do this. So essentially, if your mitochondria is there's a breakdown in the energetic process of the mitochondria, right? So pancreatic cells cannot release insulin. There's a high blood sugar level, right? And
mm,
then you have type 2 diabetes. Even type 2 diabetes is nothing but your ROS. There's too much of energy resistance, the high production of ROS, which is impairing the translocation of GLUT4, which is required to transport the glucose from bloodstream inside of cells, right?
mm,
So it's an
mm, mm,
adaptive response, as I mentioned, right? So it's an adaptive response of cells because cells is too much overwhelmed with a lot of this high energy resistance. It can be high supply of electrons, which could be because of ghrelin, right? High ghrelin or low
mm.
leptin, right? Or probably there could be a lot of inflammatory cascade which could, reduce the flux of electrons.
So, now that you mentioned about the part where uh, the damage in the processes related to the mitochondria is going to impact your health, right? Now, let's move to the part where we at Gene Pitletics, how can we measure all those functions?
Okay, so right now, as we speak, what we are doing is that we are measuring the crosstalk between microbiome and mitochondria, right?
Mm,
So we are measuring, first of all, we are measuring that what your gut and oral microbiome are secreting, what metabolites they are secreting. We measure thousands of them, right? And how those metabolites directly disrupt the flow of electron in the electron transport chain, right?
mm,
So, for instance, precursor, TMAO, LPS, all these can reduce the flux of electrons, which can increase the high energy resistance, which can lead to high energy resistance,
mm,
which could lead to production of ROS, right? Which
mm,
again can tell your they can leak out their DNA or they can communicate to nucleus to produce cytokines, which tells other mitochondria of the possible threat, and then all other cells, even including the brain cells, get into energy conservation mode, right? So to
okay.
conserve energy, so that the immune cells can respond to stress. Right? So ideally, that's a collective decision making by all the cells taken together. Right?
Mm,
So all cells are
mm,
bound. So you don't understand this. All cells work in a distributed network. Right?
mm, mm,
So every cells communicate and then they work together towards growth and development and health of our body as a whole. Right?
mm,
And that is
mm.
what cancer is, right? So essentially, if one cell detaches from the cell collective, right, that is what cancer is.
Oh, now I get it. Because it's, earlier people used to say all kind of wild things about cancer that, it's a genetic mutation which causes it. And yet these things have no bearing until there was a research which I think in one of our talks with Dr. Thomas Seyfried, he mentioned previously that, most of them don't have a cancer
I'm
mutation.
going
It's
to
a
add
genetic
to that
mutation.
because what Professor
Yes.
Thomas Seyfried said was that it is a
Yes.
mitochondrial metabolic disease. Yes, it is, right? But what happens is
Mm,
that you have to understand that, you know, there are two cells. So let
mm,
us go back to the bioelectric patterns, the voltage gradient, right? So we have to
mm,
maintain that voltage gradient, which is maintained by the ion channels. Ion channels
mm,
reside inside the mitochondria, right? And when
mm,
there's high ROS, right, there's a calcium
mm,
overload, right? There's a
mm,
there's a disruption in the calcium currents. Okay,
mm,
right? And when there's disruption in calcium currents, they become an sort of you know a barrier in the gap junctions. Gap junctions are
mm,
the way we communicate, right? With
mm,
uh,
mm,
we communicate, two cells communicate via gap junction. When so these
mm,
high iron overload, high calcium overload, right?
mm,
The disruption in the calcium current further blocks this gap junction, right? So it become an
mm, mm,
electrostatic barrier in the gap, and then cells cannot communicate. So cell which cannot communicate with the cell, it actually starts pursuing its own goals. And so every cell has its own goals, its own objectives. So the objective is to, I don't know about tomorrow. I have to grow right now, so I have to grow in the next 2 minutes. So they start fermenting glucose and glutamine because the cells have detached from the collective. They fall back on the pre you know symbiotic era where they are proliferating, they are falling for fermentation pathway when there was no oxygen environment, right? And they start fermenting
mm,
glucose and glutamine. So again,
mm,
epigenetic modification or mutations are downstream phenomenon of this calcium overload and this breakdown or dysfunction of the calcium currents,
mm, mm. That's
or
right.
what we call is the transmembrane potential of the cells have become depolarized.
Mm, mm,
They have
mm,
become positive,
mm.
right? And that's what cancer is.
That's right. So, Sushant, we've now come to the conclusion. We've
Mhm.
almost come to the end of this discussion. Any last words would you like to add here?
First of all, genes does not define your destiny, right? Your genes are static. Your mitochondria controls your epigenetic modifications, right? Your
That's right.
mitochondria is much more than a powerhouse. It is a motherboard of your cells. It controls every aspect of your biology. Mitochondria is a distributed network. They communicate. They are communicating every single second, with the mitochondria, other fellow mitochondria, and they communicate about their
Mm,
current state of affairs, and that's how they try all are working together to bring the body into homeostatic state. So probably that could lead to a new allostatic state. That could lead to, so if you have high blood pressure, hyperglycemia, headache, all these are the allostatic responses to the stresses mitochondria is experiencing, right?
mm,
So you have
mm,
to fix your mitochondria, and without energy, without flow of energy, you cannot survive. That's the basic,
mm,
right? It's a basic of this
mm,
thing, right? So rather
mm,
than, because I know the pharmaceutical companies are focused on the DNA, RNA, protein drug pipeline because that's a typical way of making money, because you identify a protein, you identify a molecular target, and you try to develop a drug that could target that molecule. But the problem is not molecule. Problem is not that hardware, right? Genes are hardware. The problem is software, which is again the bioelectric patterns. So in the bioelectric patterns, there are a lot of these, memory is stored, information is stored, right? And a lot of anatomical, blueprint is stored, that how your body structure should be, how to drive regeneration of your cells, regeneration for body organs. So if you if you cut your liver, probably liver regenerates, right? It is your genes cannot do on its own. So consider this, your genes as a bricks, right? So
mm,
these bricks can be used to form building, right? But genes cannot do on its own, right? So ideally, genes cannot build building on its own. So there are bioelectric patterns, right, which reads the information, which reads the anatomical blueprints, and then they use the information, right, to build. They can even override these genetic instructions, right?
mm,
So we can, just by
mm,
altering this voltage, potential voltage gradient, we can even override the genetic instructions, right? So ideally,
mm, mm,
mitochondria is and again, you have to understand how your microbiome communicates with the mitochondria, how your external factors, environmental factors communicate with the mitochondria. Right? If you
mm,
are thinking bad, if you are thinking, if you have negative thoughts about someone, that directly gets into inside your cell, directly impacts your mitochondria. That is also a stressor for the mitochondria.
Of course, that's like the energy mechanism.
Hmm.
Whatever you manifest, these things they kind of impact your own biology as well, right? So in the energy medicine, it is said that whatever you manifest, your thoughts are, your kind of personality are, these things matter a lot.
And another
Yes, so
thing I
I
want to
can
add before we sign
relate.
off. So from a cancer perspective, I want to add because this month is again an oral cancer awareness month, right? So we
Mm.
would be doing a lot of talks and videos on oral cancer, right? But it is very important to understand that it's a very wrong approach of killing cancer cells by using chemotherapy drugs. You
Mm.
then the approach should be that cancer is detached from the cell collective. So how do you reinstate those signals, reinstate those ion channels so that the cell can rejoin the cell collective, the affected cell can
Mm.
rejoin the cell collective, right? So ideally
Right,
there is an cooperation between the cells because every cells is learning from other cells, metabolic experience and their past events, and then they are working
right.
together, right? So all cell work together to keep you healthy. A single cell cannot do it, right? And the single cell is more concerned about its own selfish goals, right? So this is
That's right.
something very important to
I think
remember.
this has been a very brilliant discussion today, and folks, I also wanted to announce that on 24th of April, we are having an exclusive web conference with some really good industry leaders where Gene Physetics is posting the importance of oral health and incorporating the oral microbiome for healthcare practitioners, right? How can they improve their
As
practice?
well as
And
mitochondria, as
and
well as
yes,
mitochondria.
of course, mitochondria part is also there in that, and we will be giving a live demo of our platform during the conference. So stay tuned and keep listening to us. Be sure that you go to the to our webinar page and do register for this upcoming web conference. It's going to be very interesting and very insightful for health coaches, healthcare practitioners, even companies who are into health tech and direct to consumer. So it can actually redefine their practice.
In that, I'm also willing to have other potential competitors who can come and join. We have no problem.
Yeah, right. So thank you folks for listening to us time and again, and we will be back soon with with another episode. Thank you, Sushant, for
Thank
such
you.
a brilliant discussion today, and thank you.
Thank you. Bye bye.